German monk who was born in Moravia, now the Czech Republic. And he was intrigued by the mechanisms to how certain plants from the same species and same type would develop atypical characteristics. So the very first experiment he did because one very prominent theory of evolution at the time was the Lamarck theory, that whatever you do during life will transform the character and the importance of the environment. So he transferred a plant from the same species with atypical characteristics next to a plant with the typical characteristics to see if there was any impact on either of them to really control for the environment, and he did not find any difference. Then he spent a year back crossing, which was not back crossing at the time, but to build non-hybrid wines for his peas. We'll get to that in a second. So they had constant characteristics, so they were pure wines. He then had the insight also to protect their flowers from exterior pollen. And by the end of his experiments, he had studied more than 10,000 plants, which is a staggering amount considering the time. And another huge factor for Mendel's success was also that he was very fond of statistics. And if he hadn't applied statistics to his experiments and tried to guess what would be the expected results from his crosses, he would not have discovered his laws of heredity. And we'll see the impact of that because Darwin was not as big on statistics, and he had actually made similar experiments but did not understand their significance. So why did Mendel choose the Pisum sativum, the pea that he chose for his experiments? We'll do the same in a few minutes when we get to Morgan and Drosophila for his experiments. It's important to understand why certain organisms were selected at a given time. So the Pisum sativum is apparently, I never tried it, I don't really have a green thumb, but apparently it's very easy to cultivate. When you cross, it gives rise to a very, very numerous offspring. They will self-fertilize and also cross-pollinize. And they had several characteristics that could be studied. So he studied 7 of them. Whether the peas were actually round or wrinkled, the color, whether it was yellow or green, the color of the seed, whether it was gray or white, the shape of the actual shape of the pod when it's ripe, whether it's full or it's constricted, the color of the pod, the position of the flower, and the length of the stem. And he came up with actually 3 laws, which he published in a local journal, which was the reason why it never got resonance it should have had. The first is the law of segregation, according to which the parent has two copies of a trait or a factor, which segregate, and then when you have the fertilization, the offspring receives 1 copy of each from each parent. His second law was the law of independent assortment, where he showed that 2 traits or more rearrange independently during production of gametes. Now, Morgan will show that this is not entirely true, but it is still true for traits that are on different chromosomes, certainly, and far away on the same chromosome. And the law of dominance, where when he crossed 2 distinct breeds, pure breeds, only one of the characters would appear on the first filial generation. In this case, when crossing the purple with the white flower would give rise only to purple flowers in the filial generation, so the purple trait is dominant. Now, the laws of Mendel with time, there are things that he could not have been aware of. One of which is that certain loci, in his case, all the loci had two alleles. But certain loci certainly have more than two alleles, which is true for the ABO blood group, the HLAs, where there are 23 alleles. And also, some alleles show codominance, which is also true for the ABO blood group, where you can, if you carry a copy of the A allele and a copy of the B allele, you won't be either A or B, but you will be AB. And there's also this concept of incomplete dominance. If you have two copies, when two copies of gene give a more intense or more severe phenotype. So in this case, two copies of the collar gene give rise to a red flower, no copies of white. And if you heterozygous, if you have one copy of the collar genes, you get pink flower. So that was Mendel. Unfortunately, Mendel's career was cut short because about 2 years after he published this, the abbot from his monastery died, and he had to take charge of the monastery, so he completely stopped doing any genetic experiments. And his theories stayed dormant in this local journal for about 35 years until they were rediscovered around 1900. And that was about the time at which, so while Mendel was aware of Darwin, Darwin was completely unaware of Mendel. And Darwin obviously is extremely famous for having been the first to recognize that you can get that transmission of variability from an ancestral population to a population that comes from that ancestral population can give rise to a completely different population. At the time, there was resistance to his theory for two reasons. First, there was very little actual proof of natural selection since then, and since the early 1900s, there's more evidence, obviously. And Darwin could not come up with a mechanism for the transmission of these characters. As I said previously, Darwin actually had obtained experimental evidence and ratios that were comparable to those that Mendel obtained, but he could not extract the significant information from those ratios. As I said, Darwin was in the camp of pangenesis, and he also thought that traits that would be acquired during life, so acquiring a scar during life, could be transmitted to children. In a sense, this could be thinking of epigenetics before its time, but obviously we were not there yet, so we can't really give him credit for that. And it's only from the 1920s on that the role of Mendelian heredity in evolution became clear. So after that, we really entered the era of molecular genetics. In 1900, as I said, 3 people actually independently rediscovered Mendel's law, and also simultaneously rediscovered Mendel's original paper. It was actually an American called Spillman who actually did the same experiments. In 1902, Boveri and Sutton would propose that hereditary factors could be responsible for Mendelian laws and could actually be carried by chromosomes. And Sutton had observed that the number of chromosomes actually decreases in gametes. In 1905, 2 individuals, Nettie Stevens and Edmund Wilson, would describe independently that sex is also determined by sexual chromosomes. So the role of chromosomes was becoming more and more better known. In 1908, Archibald Garrod was the first to propose that the concept of inborn errors of metabolism. Obviously, hemophilia had been described in the 900 some, and some people had described. So we can't say that he was really the first to describe genetic disease. Garrod was interested. It's an interesting story. Garrod was actually fascinated by families where are individuals who had alkaptonuria. And alkaptonuria sounds very bad, but is actually a benign condition where urine, once it gets in contact with air, turns dark brown black. So it actually has absolutely no consequence on health, but it is very striking. And he recognized that while this was exceedingly rare in general, in people with consanguinity in the family, it was actually much more frequent. So inborn error. So he guessed that genetic abnormalities could be responsible for errors metabolism, and he actually has a tetrad. He studied also a couple of other diseases, including albinism, over his career. The terms of genes, genotype and phenotype were introduced in 1909 by Johansen, but people actually had no idea what the actual genes were. But now they had a better idea that chromosomes could be where these factors were located. And then we get to Morgan, who has obviously, since I work in Drosophila, I have a fondness for, who is actually the founder of experimental genetics. He won the Nobel Prize. He's first in a long line of Nobel Prize winners that I'll speak about within the next few minutes, in 1933. And like the choice of the peas for Mendel, the choice of Drosophila for Morgan was carefully planned. He chose them because they were easy to raise at a very low cost in his famous fly room. Their generation time is quite short, 10 to 20 days, depending on the temperature you're raising at. They generate a huge amount of offspring, and each and every female can lay about 500 eggs, but I have never had anything close to that kind of productivity, even with my modern stuff. So I don't know how they did it. And that turned out to be important also. They had giant chromosomes in their salivary gland, so you could observe chromosomes directly. Other factors which turned out to be very important in the development of Drosophila genetics over the years, but were unknown at the time of Morgan, was that there is no recombination in males, which is helpful for traits on the X chromosomes. On the X chromosome, and eventually people would develop balancer chromosomes, so you could prevent recombination and really easily follow the mutations in lines. There are very, very powerful tools to do genetic manipulation. And about 60% of the genes associated with disease have an homologue in flies, and including, the fly has a very, very interesting, tiny but very interesting brain. So the first thing that Morgan of significance that Morgan published with these flies came about when, and the legend is a little bit fuzzy, but apparently his wife found a male fly with white eyes. Usually Drosophila had red eyes. So anyway, someone identified a fly with white eyes, which apparently Morgan for the next week protected with his life. And he would cross this male with females with red eyes. We say females, and we don't know how many exactly. And in the first filial generation, nearly all the flies had red eyes. There were 3 males with white eyes, which could be either spontaneous mutations or, the white gene could have been present in one of the females that the flies would cross to. But in the second generation, 2,400 females with red eyes, 1,000 males with red eyes, but 700, nearly 800 males with white eyes. And he was trying to fit that with the concept of Mendelian transmission, and it did not fit, except if he considered the sex chromosome. And people had the concept that males were XY and that females had XX. And then Mendelian principles still held up. Over the next couple of years, Morgan would identify more than 24 mutations, which would help give rise to two different, also very important concepts. So the very next year, he discovered the concept of genetic linkage and recombination. So Morgan would observe that the parental phenotypes, when he was doing his crosses, that the parental phenotypes were overrepresented in the offspring, which suggested that in certain cases, for certain crosses, certain traits tended to be transmitted together and not completely independently as Mendel had stated. But he also noted that these traits that were often transmitted together at times would be separated, and the linkage would be broken. And he also noted that certain traits that were on the same chromosome, because they were starting to be able to build a map. Well, and it was easier for the X chromosome, obviously. They were trying to build a map that certain traits that certainly were on the same chromosome were not linked, never linked. So he proposed that there could be, that there was linkage, and he proposed this concept of recombination, which was his original drawing here, where they knew that chromosomes would combine, that paired chromosomes could combine, and then he thought, well, maybe it's because they recombine and they exchange information. And he had the idea that the frequency of recombination was probably due to the distance between these two genes. And when you had recombination, it would break the linkage between the genes, which led to another very important discovery in his lab, which was done by Alfred Sturtevant, a PhD student in his lab, in 1913. And Sturtevant understood, so from that concept that, probably the frequency of recombination was related to the distance between two genes, Sturtevant thought, well, maybe we can physically map these genes if we know how frequently, one to another, if we know how frequently they recombine. And indeed, it was the case. He spent many evenings with a ton of data to try and figure out the distance between these the different factors. And he would use the proportion of crossover as a measure of distance between the two factors. So here is what pretty much one of his first maps was. The very first map was the map of the X chromosome, but here is some data that he had on the second chromosome. So he gave a linear position to all the traits according, and all the genes according to the frequency of recombination. So if there was a 70% chance of recombination between the body color gene and the vestigial gene, the distance was 17. And which was interesting was that it would fit that the distance between the body color and the eye color gene was 8%, and that between the eye color gene and the vestigial gene was 9%. 9 plus 8 is 17, and it did fit. So one unit was 1% of recombination. Eventually these units were called centimorgans, to honor his supervisor. By 1915, Morgan did write up a paper where he would summarize the main tenets of his theory. And there he said, he'll soon call them genes, but he'd say distinct pairs of factors localized on chromosomes carry hereditary information, and they segregate in germ cells and combine during repetitive reproduction. Certain characteristics are linked to are sex linked because they are on the sex chromosome, on the chromosome that determines sex. And certain characteristics are sometimes associated because they are close to one another, but then this linkage can be broken by recombination and crossover. So we stayed in Drosophila still, because in 1927, Hermann Müller, who was an American, was able to induce, it was the first to be able to induce and actively manipulate the genome of another organism, or an organism, using X-rays in Drosophila. And using that, he was able to generate, well, they had generated, a few tens of, or had identified a few tens of naturally occurring mutants over the previous 15 years. Mueller was able to generate hundreds of mutants very, very quickly, which also proved to be very useful. So Mueller also won a Nobel Prize, and Beadle and Tatton also shared a Nobel Prize. Beadle was also a Drosophila worker who had trained in Morgan's lab, but his main experiments were actually done in Neurospora, and he switched from Drosophila to Neurospora because he wanted to really look at very, very basic principles, and he needed an organism that would replicate very, very quickly. So if we take a second, because they won the Nobel Prize because they were able to demonstrate and clearly link how enzyme synthesis was associated because how genes were able to command the synthesis of enzymes that are very important for a metabolic process. And that was not known at the time. So what they did is Neurospora is actually can be haploid, and the spores are actually haploid, and they can be deployed, not deployed, but anyways. So the haploid spores, they actually irradiate, and usually they grow on a very minimal medium, a medium without any amino acids. So they would irradiate the spores and then cross them with the wild type Neurospora. Then they would raise them on a complete medium and then test them in minimal medium. And some individuals would not grow on the minimal medium. So they were mutant and some metabolic process was interrupted. So what they did was go back to that complete medium, now then use minimal control and a complete control, but also grew them on a minimal medium, but with each and every tube was supplemented with one amino acid. So in most of them, they did not find any growth, but if they were to find growth in the one where they had added arginine, then suggested that the metabolic process that was affected had something to do with arginine, the production of arginine. So they were able to, dissect a lot of, not really dissect a lot of the pathways, but identify a lot of the mutants that were specific for certain the metabolism of a series of amino acids. In 1944, DNA was formally identified as a vector of heredity. Before then, and even afterwards, because even at the time of Watson and Crick, some people still believe hemoglobin was the vector of heredity. We have to step back a little bit, because in 1928, Griffith showed that if you injected strains of pneumococcus in mice, and the surface was smooth, the mouse would die. If the surface was rough, the mice would survive. People then finally understood that was because the rough surface was detected by the immune system, while the S surface would escape the immune system. But if you inactivated the S strains, so the smooth strains that would kill the mouse if they were living, nothing would happen. So the pneumococcus would not reproduce, and the mouse would be fine. However, if you injected both live but innocuous pneumococcus with dead but lethal pneumococcus, the mice would die. And this suggested that there might be an exchange of information between the two bacteria. And it took 16 years for Avery, McLeod, and McCarty to figure out. So first, Avery figured out that you don't need the mouse; you could do all that in the test tube. And both Avery and McLeod were actually Canadians from Nova Scotia, but working in the States. And what they did was basically, gradient separation, and it would isolate different cell components, including DNA, and show that if they don't add DNA to the mix, there is actually no transfer of information, which really showed that DNA was the vector of heredity. They had to purify 20 gallons of bacteria to find that out. And then we get to the identification of the structure of DNA, which completely changed molecular biology. So in 1950, Chargaff had discovered that DNA bases actually, and across species, were always found in the same proportion. Adenine and thymine were always equal. Guanine and cytosine, always equal. And people were considering the possibility of a helical arrangement, but there was no clear model for it. And Crick and Watson were interested in studying DNA because they both thought that it had a fundamental importance. Watson was a 23-year-old PhD student, and Crick was a very relaxed student. And they started working together informally, and they would build 3D models for 2 years, borrowing from the method that Linus Pauling was trying to use to find the structure of the vector of heredity. And there was a significant contribution from X-ray diffraction results from Maurice Wilkins and Rosalind Franklin. And on one evening in a pub, I think, Watson realized that pairs of adenine and thymine and pairs of guanine and cytosine actually had the same shape. And if they were linked with a hydrogen bond. And they quickly figured out that a double helix model made a lot of sense because it allowed to have paired sequence and allowed the molecule to be copied if you were to separate the strands. So and in their paper they state that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. And that is probably the most important part of that model. I encourage you to read that paper. It will take you like 5 minutes. It's written so you can change the face of biology in an evening and 1 page in Nature. And I encourage you to go and have a chat with James Watson, who is a peculiar individual who still acts as the emeritus director, even though he lost his title. He still has his office at Cold Spring Harbor. So if you go to a course or a seminar at Cold Spring Harbor, you'll see him hang posters on billboards. So 4 years later, Crick wrote another seminal paper, which was called Protein Synthesis. So there was this. So there are 2 important ideas he raised. One was the sequence hypothesis. The other is the central dogma of molecular biology. So the sequence hypothesis was that they quickly figured out that the order of bases could code for specific amino acids, and that each and every word of that code could call a specific amino acid. And Crick even went so far as to predict that with 20 amino acids and 4 bases, the simplest code would be a code of triplets. So that was he was a clever individual. And the other main concept was the central dogma, which he took a lot of flack for, actually using the term dogma, because it's well, yeah, he took a lot of flack for that. So that the main tenet is that information is transmitted from DNA to RNA, then to protein, but never from proteins to DNA. And we know that now, proteins can actually significantly modify DNA significantly. So we were still in the era of molecular genetics, and now it's just a list of Nobel Prize winners. Kornberg would crystallize DNA polymerase. In 1960, Sidney Brenner, who won a Nobel Prize later for his work